Right. Sorry about that interruption. So advancing to the next slide, a little bit of overview of the computational modeling and simulation project within MDIC. MDIC is a 501c3 public-private partnership. We're a member-driven organization, and one of the projects our members have focused us on is computer modeling and simulation, with the goal of quick and predictable access for patients to innovative technologies through the use of objective evidence generated from computational modeling and simulation. This is a long-term activity with some opportunities for short-term wins, and today we'll talk about some of those future visions. The project at MDIC has an organizational structure with a board champion, Randy Schistel from Boston Scientific, the Global Vice President of Research and Development, myself as Program Manager, and Kyle Myers as the FDA PI for the Computer Modeling and Simulation Project, and she's the Division Director of Imaging Diagnostics and Software Reliability. Our steering committee has over 23 members from 15 of our member organizations, and we have 7 working groups within the Modeling and Simulation Project. Those working groups conduct the work that the project identifies, and today is focused on the heart vasculature working group and some of the things that they see on the horizon in the future. As a quick glimpse of our Modeling and Simulation Steering Committee, we have representatives from the medical device manufacturers, the FDA, as well as nonprofits and expert software organizations. So a nice blend of expertise at the steering committee level. And with that, I'll hand over the talk today to Wei Gan, who will be working as our MC. He's a PhD and has PMP certification, engineering manager of modeling and simulation within Medtronic. Wei, thank you.
Thank you, Dawn. Good morning and welcome everyone. And thanks for joining us this morning. And the MDIC Computer Modeling and Simulation Project has several working groups. The Heart and Vasculature Working Group is one of them. Today's telecon is hosted by this working group. Next one. And the working group works with several other working groups as well as all stakeholders to increase the use of modeling and simulation as evidence for safety and efficacy. And this will be a journey, as you can imagine. This can be achieved by developing tools, process, and methods, along with encouraging basic research so that we have credible models to simulate virtual device and virtual patients. Next one. The Heart and Vasculature Working Group is chartered to demonstrate the potential of modeling and simulation by simulating clinical relevant use conditions, especially in the heart and the vasculature space. And there are many activities ongoing, and some of the key deliverables include hosting MDIC series telecons like this one today, as well as we're going to distribute a request for information so that we can learn what are the models out there, especially related to heart and vasculature and anatomy and physiology. And going forward, the working group will be working together in pre-competitive space to demonstrate the potential for virtual testing using those kind of models. The first example could be the virtual lead testing in the intracardiac lead fatigue type of study. If you are interested, please contact Dawn or myself so that we can include you if you are an MDIC member. Next one. Today, we have 4 distinguished speakers. Richard Gray, as well as Pras Pathmanthan, are scientists at the Office of Science and Engineering Laboratory in FDA. So Rick has been studying heart functions over many years, and he is expert in cardiac and electrophysiology. He has been doing extensive study in the testing and the modeling side, especially with focus in fibrillation and defibrillation type of study. Rick has a bachelor degree from the Bucknell University, as well as a master and PhD degrees from University of Virginia. Rick, after his study, he went to the State University of New York as a postdoc in the medical school, as well. And then after that, he has been a professor for a decade, more than a decade, at the University of Alabama in Birmingham, where he is also a director of graduate programs. Plus, it's also has also done intensive research in the cardiac space, and he had studied at both Cambridge and Oxford University. He has a PhD in computational biology and got that degree in 2007. He had a particular interest in whole heart modeling, as well as the verification, validation, and uncertainty qualification of those type of models. Plus, is a member of the MDIC Computer Simulation and Modeling Simulation Steering Committee. Marc Horner is from ANSYS, and he has a chemical engineering PhD from the Northwestern University. He has been doing intensive professional services for biomedical clients in the past and right now as well. But at this time, and he also has the responsibility to coordinate business and technology development for the healthcare sector for ANSYS. Thierry Marchal, he is the industry director for the healthcare sector for ANSYS. He likes working with all clients and getting all the feedback and information to drive medical device strategy, so that he can help the company to deploy engineering simulations and accelerate product development for the medical device manufacturers. He has also worked with Fluent, worked for Fluent as a materials market segment manager. He also, as well as a brief working experience for Polyflow. It's also a brand of ANSYS. Thierry has a BS and MBA from Catholic University of Louvain in Belgium. Without further ado, I will hand it over to Marc and Thierry, and so that to hear their story about the in silico clinical trials. Here you go, Thierry and Marc.
Thanks, Wei, for the introduction. I appreciate that. Okay, so next slide, Don. Okay, so just here's an outline for our presentation today. First, I'll open with an introduction to ANSYS and our motivation for joining MDIC. Then we have 3 examples of in silico clinical trials and physiologically realistic simulations that we'd like to share today. These are by project name, the AngioVision Project, the IRIS Project, and the Fluid Initiative. And then we'll wrap up at the end with some conclusions. Just so you know, I'll be presenting through the end of the AngioVision Project, and then my colleague Terry will take over and present the second half of the presentation today. Next slide. Okay, so just to give everyone a brief introduction to ANSYS, we were founded in 1970. We develop engineering simulation software that can be used to perform what we term here at ANSYS simulation-driven product development. For those of you in today's webinar who aren't familiar with engineering simulation, these tools enable product development teams to perform virtual experiments or simulations on a computer. These simulations are based on first principle conservation laws, which means that when performed correctly, a computational model can predict the real-world behavior of a medical device or any other product. Each of the boxes on this slide summarize some of the capabilities of our physical modeling disciplines, which are mechanical, fluid dynamics, thermal, electromagnetics, and systems-level modeling. All of our software tools are available through a unified platform called Workbench, which enables multi-physics and multi-scale parametric analysis and design optimization. And it's the depth and breadth of our product portfolio and the interconnectedness of our tools that's a primary reason why we maintain a significant presence in so many different subsectors of the healthcare industry. And, you know, one final point here on this slide is that our membership in the MDIC is driven by the desire of our many customers in the healthcare market, not only to advance their simulation capabilities for their product development, but also to elevate the regulatory significance of their modeling efforts. So we definitely see MDIC as an important partner in that way. Next slide. So now on to our first example of physiologically realistic simulation, which is the AngioVision project. So AngioVision is a collaboration between ANSYS, who provides expertise in mechanical simulation, the University Hospital in Rennes, who's the clinical partner, and TheraVA, whose end-of-size software tool is used for planning endovascular procedures. This slide shows a screenshot of the end-of-size software environment. So after importing a patient CT scan, the end-of-size software provides a surgeon with an analysis of the patient's anatomy. In this case, we're seeing a summary of a patient whose arterial wall has degraded to the point that there is now a bulging in the region of the aorta just below the vessel wall, just below the kidney. And, the risk of death to this patient is very high if the vessel wall ruptures. Therefore, a device called a stent will be implanted to reestablish the flow path. But stents come in many shapes and sizes, so one of the many critical decisions a surgeon has to make is which stent is appropriate for a given patient. End-of-size software has proven to be quite valuable in this aspect of decision making, but there is still a need for going even deeper into the planning phase to predict the interaction of surgical devices with the vasculature. So here at ANSYS, as part of our membership in the AngioVision project, we've been working to amplify the TheraVA software through the co-development of an FE module that can predict device interaction with soft tissue. Next slide. But you might be wondering, what are the clinical challenges associated with these procedures that would require mechanical simulation? And one of the key factors to keep in mind is that we have rigid medical devices such as a Guidewire interacting with soft tissue. These devices may lead to significant deformations of the vasculature during the procedure. So the example on the lower left-hand side, we see a very complicated anatomy, and in this case we have a patient with a kink in their left iliac artery. This arterial segment will have to migrate up the length of the Guidewire as it's introduced. On the flip side, another option could be that the Guidewire actually pokes through the iliac artery, and that would be catastrophic for the patient. Therefore, physiologically realistic mechanical analysis can help the surgeon design a successful treatment plan. Next slide. So this slide here gives you a workflow for the AngioVision from the mechanical analysis perspective. So the inputs into the model are the patient anatomy and the information about the medical device or devices. And for today's discussion, we'll focus in the first part of this talk on the Guidewire itself. So following the workflow, we have a Guidewire and patient anatomy that are combined into a single model, and a computational mesh is created. Next, we go into an iteration loop to determine the appropriate boundary conditions and material properties for the model. In this case, a series of simulations are conducted until the Guidewire location predicted by ANSYS matches the location of the Guidewire determined from images of an actual patient undergoing a surgical procedure. We go through this loop repeatedly to minimize the error in the predicted versus the actual Guidewire location. Next slide. So now to give a little more information about model inputs. First, we rely on end-of-size software to construct the patient anatomy, which in this case provides the vessel centerline location, along with splines of the vessel shape along the length of the artery. We use this data to create the surface model shown on the far right-hand side of the slide. Next slide. And we can extract other important information from the scan data as well, such as the variation in arterial wall stiffness and the proximity of the abdominal aorta to the spine. And this latter piece of information is important because the spine acts as a rigid obstruction to artery wall deformation when devices are introduced. Next slide. And lastly then, in terms of inputs, we need to incorporate engineering information about the tools that are being modeled. And again, just as a reminder, today we're talking about the geometry and mechanical properties of the Guidewire. Those are shown on the left-hand side of the slide. Next slide. So the simulation of the interaction between the Guidewire and arterial vessel wall can be accomplished in one of two ways. One's by modeling the insertion directly, or a second option, a slightly different approach would involve allowing the device and the aorta to come into mechanical equilibrium with each other. So we're currently investigating both options, direct insertion versus this mechanical equilibrium approach. And for today, I'll just show you results for the mechanical equilibrium simulations that we're doing. And the first step in this process is to position the Guidewire at the insertion point We then used the centerline information provided by EndoSize software to position the guidewire along the vessel centerline. This step introduces strain into the guidewire, so when we release the guidewire, the vessel and guidewire can come into mechanical equilibrium, providing a prediction for the patient-specific location of the guidewire. And that's what you see on the right-hand side of the slide. Next slide. So the results of analysis like what I showed on the last slide can be analyzed for centerline displacement along the length of the anatomy. In the picture on the right, the blue-colored artery is the location of the patient anatomy that we took from the CT scan, and the red-colored artery is the final location after the mechanical equilibrium simulation. And what we can see in the XY plot on the left is that the displacements in the iliac are much greater than those in the abdominal aorta. So one conclusion here is that we can't neglect what's happening in the iliac when we're tuning the model. Next slide. So this displacement information can also be used to validate the model. In this case, validation involves the quantification of two sources of error, registration error and simulation error. So registration error is the distance between the preoperative and intraoperative center lines. This error occurs because the vasculature does not stay rigidly in place over the 2 to 3 week delay between the acquisition of the preoperative planning scan and the surgical procedure. So you see, a representation or an image of a registration of a planning scan, and then this, surgical day scan. And what we see is there has been some motion of the arterial vasculature over the planning time. Simulation error then is the distance between simulation results and the vessel center lines measured during the procedure. Next slide. So this slide shows the results of a clinical validation study with 18 patients. In this case, 18 patients were divided into two groups. Group A consisted of 10 patients whose information was used to train the model, and Group B patients were the 8 patient data sets used to validate the model. Registration error was around 1.6 millimeters for Group A and decreased to 1.3 millimeter for Group B. The total registration error for all 18 patients was 1.5 mm, so you can see that in the top row of the table in the middle. Simulation error increased slightly to 2.1 millimeters for Group A and then 2.8 millimeters for Group B, with a total error of 2.3 millimeters. So, what we see is that after tuning, there was a 33% increase in the error for Group B, but in light of the fact that no tuning was performed for the Group B patients, we believe this is an excellent, example of physiologically realistic simulation. Next slide. So the next step in the AngioVision project is to model deployment of a stent graft to the vessel wall, again with the goal of providing surgeons with additional information about the potential success of a procedure during the planning phase. Next slide. So the methodology in this case is similar to what was done with the Guidewire simulation, which is to position the stent in the patient anatomy and then allow the anatomy and stent to come into mechanical equilibrium. Next slide. This slide shows the shape of a stent graft extracted from one of our study patients, superimposed on the stent graft shape predicted by the mechanical simulation. And so we're not yet to the point of establishing quantitative agreement like what I showed with the Guidewire simulations, but we can already see here that there's good quantitative agreement between the mechanical simulation and the clinical outcome. So that's the end of my part of the presentation. I'll now turn things over to my colleague Thierry Marchal, who will provide 2 more examples of how ANSYS tools are used in physiologically realistic simulation. And what's really exciting is it's to the point of even guiding patient selection in clinical trials. Thierry, over to you.
Okay. Thank you, Mark. I hope you can hear me okay. Good morning, everybody. And beyond the use of simulation during the surgery planning, as illustrated by Mark, there are clinicians who feel the need to use simulation results to assist them directly in the operating room. So this is the case of the team of Dr. Kostala, Gascu, and Professor Bonafé from the Hospital of Montpellier. In the next slide, you see they want to treat cerebral aneurysm through a minimally invasive treatment introducing a web into the aneurysm sac. So this is much faster than packing coils, and this avoids to have cerebral stent in the artery wall itself. But if the web is too big, its deployment could induce some large stress in the aneurysm wall, possibly leading to the rupture of it. If it is too small, it might fall and obstruct the artery. So clinicians would welcome some guidance to select the position of the web, to select the web and the position of it, and they believe over there that the simulation can be very helpful over there. But the key was to make sure that they can validate simulation first. So on the next slide, the idea was to simulate the process quickly, in less than 1 minute, so that, if necessary, this could be done in the operating room. So for that, a shelf equivalent model was developed for modeling the web itself. So here, you can see that the first patient, for him, they built an in vitro model to select the web, or more exactly, to validate the simulation-driven selection of the web before implanting it, giving some very nice correlation between the in vivo, in vitro, and in silico approach. The next slide is showing the second patient, for which the in vitro approach was not considered anymore. There is a good qualitative validation, and this is illustrated by the tip of the web, shown by the blue arrow both on the in vitro and in silico result. This also suggests that part of the web is slightly entering into the artery, what could impact a little bit the blood flow, which is not ideal. The next slide shows the third patient, for which a bigger web was implanted. And once again, this was correctly predicted by the in silico approach. In total, 7 patients were successfully implanted in the first part of the project, combining in silico guidance and in vivo validation. The next phase of the project will include clinical validation with more than 30 patients. For that, the clinicians were requesting to have a specific vertical application. So the next slide presents the vertical application that they are currently building to help the clinician to take advantage of this tool during the operating room. So you first see the import of the patient geometry. Next, you can see that the selection of the web model from an existing database. Next, the surgeon can position the web, and the deployment of this web for this patient at this location is then calculated. And hopefully we'll get the slide back very quickly. And so after just a couple of minutes, so maybe less time that we need to get the slide, the surgeon will be able to see if the web location was okay or if it was too dangerous for the patient.
So Thierry, it's on the FRI slide right now.
Okay, good. So here you will see the last example coming from the respiratory industry. So Fluida has developed what they call the functional respiratory imaging, or FRI. Can you come back to the previous slide? Well, no, that's okay. Let's stay here. The goal was to accelerate the clinical research and clinical trial. So here, this slide explains how it works. We classically start from a scan to get a computed tomography, so a CT scan of the lungs at the full respiratory capacity and residual respiratory capacity. So a quasi-automated process allowed to create the 2 patient-specific geometry and to model the airflow. So on the next slide, you can see that by comparing the 2 geometry, you can calculate the local variation of the volume of the lungs or the local resistance of airway, as illustrated here. Getting some local information is essential, because if some part of the lungs are infected, for example, they won't work, but the rest of the lung will compensate for that, so that you will not notice the early warning of the infection and not being able to treat it accordingly. So this is not the only way to do that. And the next slide compare this in silico approach with the well-known FEV1, the forced expiratory volume in 1 second. And you can see that there is a good correlation both for airway resistance and airway volume. But first, FEV1 is a global measurement and therefore doesn't allow you to detect some local infection or local problem in the lungs. Second, this measurement has some noise, and the noise is coming from the fact that it's taking into account both the lower airway and the upper airway, like the mouth and the throat, which can perturbate the impact. Also, it's taking into account the condition of the patient itself, like the age of the patient who cannot perform the right effort. But this is not directly related to his respiratory system and airway volume. So as this is what we want to test. So for a given value of the FEV1, you can have different value of the airflow resistance or the airflow over volume. If you want to make sure that you cover all the possible airflow resistance, airway resistance, and airway volume, you need to get a large number of different patients. By better understanding the noise over there, you will see on the next slide that if you know this noise better, you can cover the desired range of respiratory capability. Can you move to the next slide, please, Don? So over there, you see that by better understanding the signal itself and minimizing the noise coming from the external capacity of the upper airway, you can reduce the number of patients that you need from 68 if you are using a classical spirometry down to 25 if you are using in silico approach in order to drive it. And this would be even more important because the next slide is showing that for chronic obstructive pulmonary disease, where the noise could be even more important, previous one, sorry, where the noise could be much bigger, the number of patients can be reduced by a factor 5 when you are taking advantage of this approach. So far, this method has been successfully applied to about 1,600 patients in order to drive some clinical testing, especially for respiratory drug delivery. Finally, I'd like to mention another approach here on the next slide, is that the same approach can be used both for preclinical animal testing and clinical testing under the human. By doing and combining the same approach for all of them, this is a nice way to accelerate the process to get a proper respiratory drug delivery process in the entire environment. So let me conclude on the next slide. These three examples have illustrated the medical world is entering the in silico area. Why now? Probably because the computational capability right now and the emergence of this kind of multi-physics system-level platform is allowing us to model a very wide range of treatment. In addition to that, as shown by Marc, the smooth workflow going all the way from patient through medical imaging and to engineering simulation is simplified the way to make some modeling for the large cohort of virtual patients. And most of the in silico publication right now are considering a very large number of patients. And last thing which is very important is that all the three presentations are directly working with clinicians. The clinicians today trust clinically validated simulation. Not only they trust it, but they're asking for them. They're asking to use it. So as a result of all of that, I would say that in silico clinical trial is really becoming a key component to traditional approach in order to accelerate the device innovation process. I would finally mention that more literature is available for each of these examples, and each of them will be detailed in a full webinar. So let me know if you want to know more about that or being invited to one of these seminars. Thank you, Don. Back to you.
Thank you, Thierry. And I think Rick and Pras are up next.
All right. Thank you. This is Rick. I'll be the primary speaker and Pras is on the line, and we'll both be available for question and answers at the end. Next slide, please. This talk is going to be much more high-level, mile-high view rather than specific modeling details. Those, most if not all of what I'm talking about has been published or is in press and can be hopefully accessed by everyone who's interested. These trees are a little blurry, and maybe that's appropriate given what they represent. They represent many decades worth of work in the academic, mostly the academic community, in the development of whole heart models, mathematical models. And they really have grown out of three different disciplines. One, of course, is the mechanics, two is the electrophysiology, and the third is the fluid flow. And the fluid flow I have at times two to indicate that there is both the fluid in the ventricular cavities that is pushed through the aorta and the rest of the circulation, but there's also the coronary artery and venous blood flow that also is modeled. So these trees just represent, and the fact that they're blurry is that there's many different approaches, but I did want to stress that they really grew out of specific disciplines. Next slide, please. And it's like the blind man with the elephant, is that the heart is the heart, and it works amazingly well as a pump and electrical organ, and we tend to model it discipline specific, but of course, as the previous speakers mentioned, that is changing. Next slide, please. So the coupling between these different disciplines is extraordinarily important, and I think we have seen and will continue to see a lot of advances in this area, and I think that's where most of the work is happening right now. In general, historically, what has happened is at the end of these trees, people in the different fields will tack on a rudimentary version of another discipline. So for us electrophysiologists, we'll build up a very detailed electrical model, and then at the end, we'll try and put in some rudimentary mechanics. And that's called weak coupling. The strong coupling is getting more at the mechanisms of the bidirectional coupling of what's going on in the cell. And over in the left is what's happening at a cellular level of how the electrical and mechanical are coupled. So the top is an electrical action potential. The middle is the calcium, intracellular calcium trace that is triggered by the upstroke of the electrical signal. And that calcium is then released, it's released into the main cell compartment, and that causes some binding of the actin-myosin filaments that cause contraction. And there's feedback from the length on the calcium, and it's a very complicated dynamic. And if that's approached quantitatively and correctly, that's what's called strong coupling. Next slide, please. These models are inherently multi-scale. I won't spend much time on this slide except to note that the electrical is, as I'll explain in a few future slides, is a little bit unique compared to the other disciplines in how the multi-scale is implemented. I didn't show it here, but in my lab we both do whole heart experiments and single cell experiments. And when you take a single cell out, it works in many ways like the whole heart. Of course, not the full beating heart, but each heart has an action potential, has a calcium transient, and contracts. And that's something that is important, I think, in when you start looking at the multi-scale. Some things like metabolism at first order are not multi-scale, so those are incorporated sometimes into the electrophysiological cellular models that I'll talk about in a couple of slides. And also to note that there are species difference, and so when we translate the existing data set to mathematical models, it's not as simple as one might think. The data is obviously more easily accessible from animals than from humans, and so we have to understand what the species and environmental differences are when those experiments are, under which those experiments are taken out, and the mathematical models derived from them. Next slide, please. I hear a little echo. Hopefully it's not disturbing too many people. This is just a slide illustrating what's called whole heart optical mapping, which I helped pioneer with my mentor back in the late 1990s, which was really neat because it gave us the ability to visualize wave propagation over the whole heart at a very high spatial resolution, from down to about 300 microns, sometimes less than that, so getting close to the cell level. And now I meant to say in the last slide, I'm going to focus on the electrical right now, and that's my specialty, and I'm not going to be going back to the mechanics or fluid flow, with one possible exception. So I won't spend much time here, just to say that we use voltage-sensitive dyes and isolated hearts, and then we look at the transmembrane action potentials at over 10,000 sites from the heart surface. And we often use two cameras to get two different views from the anterior and posterior side. Next slide, please, and start the movie if you would. So I'll start by just showing you what electrical activity in the heart looks like. These movies are slowed down many times This is a normal rhythm of a rabbit heart, and so the scale is about 4 to 5 centimeters from top to bottom. That's the atrial activity. The gray level represents transmembrane potential, and then the ventricular depolarization is just completed, so that completes the QRS, and then this is the repolarization phase during the action of potential plateau where it's coming back down to rest. So that's the normal activation sequence in the rabbit. It occurs about 3 times per second, and we'll move on to the next slide. Here's a slide. Don't start it quite yet. This just represents what happens when you pace the heart. The sinus rhythm is the normal rhythm is meant to generate a synchronized contraction. However, if you pace from the surface of the heart, which you can play the movie now, Don, you'll see a wave propagating away from the asteris, which is the pacing site. And so there you see that it propagates around away as an ellipse through along fibers, then across fibers. Next slide. And just the last movie, just start to play it. This is a movie of fibrillation and defibrillation. We'll probably stop it before it gets to the end. Sorry about that picture being on front. But this is ventricular fibrillation in the rabbit heart. It's not coming across nice and smoothly on my screen, but it does in the movie. There's a defibrillation shock. And I could spend a whole talk on mechanisms of fibrillation and how to analyze it, but that isn't the purpose of this talk. So let's move to the next slide. Okay, so why model the heart? I don't think I have to tell this group. You can aid in the design of medical products. It can complement and, yes, even replace decision-making in some cases. Of course, patient-specific medicine has the ability and may transform medicine, and that's where modeling will be very important. Whether we need physiological models or not it depends on the context of use, I presume. They do provide support of model predictions, and they may be required to reproduce certain behavior. Next slide, Don. And I do want to indicate that in our field, cardiac electrophysiology, the pacemakers and defibrillators and cardiac resynchronization therapy have really been driven by our deep understanding of how electric fields affect the heart and how the heart functions. So that's really been integral to the design of these devices. And so I think in this particular field, we're quite advanced in terms of the electrical physiological modeling. Okay, next slide. This is a short-term application forecast. This does not represent the official FDA view, and it's obviously incomplete. But we see it being used in near future in terms of pre-electrophysiological planning, like ablation planning. Of course, cardiac resynchronization therapy, where to place the leads, defibrillation as well. Also for assessing algorithm efficacy, like mapping algorithms. And then, of course, on the drug side, this is it's a very important aspect, because when you're when you're submitting a drug application, knowing the mechanism of action is very, very important. Next slide. I think in the interest of time, I'm going to go through a few slides. I don't need to show these examples, and probably won't spend much time on the multi-scale aspect of it. Go to the next slide, please. Yeah, why don't we just play this slide? This is on the left is unstable reentry or fibrillation, and this is a model study that we did and showed we could test a hypothesis that affecting the kinetics of the L-type calcium channel would actually stabilize fibrillation into a tachycardia. So once again, mechanism-driven design, in this case for drugs, can be very, very important. Next slide. This just represents the multi-scale nature of the heart modeling. And very important, it goes from subcellular membrane level to whole heart, and you couple both cellular dynamics and heart geometry, which we'll show. So next slide, please. And this is just a quote I wanted to put in here from actually a Nobel laureate in molecular biology, and he makes the statement that he believes very strongly that the fundamental unit, the correct level of abstraction, is the cell and not the genome. And in the case of the heart, where the cells are very, very well coupled and there's macroscopic wave propagation and reentry, I think this is particularly true. And a lot of the mathematics and modeling that goes into our field are how do the cellular dynamics affect macroscopic behavior. Next slide, please. So just play this movie. It starts with a high-resolution representation of the rabbit heart that we're using. You can see it's got very detailed structure, and the inside of the heart is not smooth, but the outside is. Here's the induction of a counter-rotating pair of reentrant waves that is happening in the 3D heart. This is a simulation, of course. And then it breaks down into multiple reentrant waves and is somewhat analogous to what we observe in the experimental laboratory. Next slide, please. So you can play this slide. One of the advantages of the 3D is we can actually look inside the heart, which is very hard to do. And what these the organizing centers of these reentrant waves in 3D are lines. So these rotating waves are rotating around lines or curves in 3D space that are called filaments. And that's what we're showing here. And if you can imagine, the mathematics and theory that goes into identifying these reentrant waves is not trivial, but it is important if you want to quantify these sources of fibrillation, which are the unstable reentrant waves. And also there's it's a very important thing to realize that the behavior is actually a very integral combination of cellular function and the heart structure, and that's something we're actively working on, as are other people. Next slide, please. So this is the verification, validation, and uncertainty quantification part of the talk, and I'll just, by one sentence, define each one, and I'll probably use the acronym VVUQ throughout. Verification: does the computer model accurately solve the underlying mathematical model? Validation: how well does the computational model approximate reality? And uncertainty quantification: how much does uncertainty in parameters of initial conditions affect the results? So very quickly, I'll try and run through an example of each and try and wrap up in 5 minutes and give us time for questions. Next slide. So verification, you want to know that your solvers are actually solving the equations correctly, which one may take for granted, but it shouldn't be taken for granted. And what Pras has done recently is developed a suite of tools, one in particular, that can be used by anyone to demonstrate the correctness of their solvers. And this can give a high confidence. You never have perfect confidence, but high confidence in the solver correctness. And by his technique, you can look at the error convergence. So it's not just the error itself, but it's how does the accuracy depend on the discrete, the time step and the space step. So that's the convergence part of it. Then I don't think I'll show the movie here, but we developed a verification test problems for cardiac mechanics solvers. And we have a whole heart high-resolution mechanic paper recently out, and part of that was the verification aspect of it. And one of that part was running 2 independently written solvers for the same problem. So that's one way to verify code. Next slide, please. Validation. This is very difficult, and it's linked to the context of use. Once I got started to try and do it. It seems like it's many different layers of the onion, so I think it's a very complex process. Next slide, please. I'll just throw a couple examples out here. One thing we do is how well do existing models represent new data, that is data that wasn't used in their development and calibration. And recently we showed that the existing models do not represent the shape of the wavefront during propagation. So a very basic feature, if you will. The models do represent the conduction speed, but not necessarily the shape of the upstroke. So one has to be very clear on what your model represents and what it doesn't. Next slide. And here, when you start talking about whole heart models, you really have different, you have the multi-scale aspect built in. So how does one validate complex models of complex phenomena that are multi-scale in time and space? Of course, I don't have an answer for that, and we're just starting to tackle that problem, but it's something that is very, very important. Next slide. Uncertainty quantification, this could be perhaps the biggest challenge in the credibility assessment. And I won't spend much time on this example except to say this is the data on the left represent the big solid symbols represent mean data from I think 16 different cells, and this data is used to construct the inactivation gate, a subcomponent of the cell model. And as far as I know, what everyone does is take the mean data and use that to construct the data set. But if you look at the cell population data, which are the thinner lines, you'll see that the mean does not represent the average cell, at least the slope of this inactivation curve. So that's something that is very important, and we showed how using different methods instead of nonlinear least squares, nonlinear mixed effects, which takes into account the population variability, and then propagated the population variability through in uncertainty. So you can propagate that through in terms of your cell model and your tissue model, which we did in a recent paper. Next slide. So I know I covered a lot, and I did it pretty quickly, but like I said, most of this is published. So VVUQ of physiological and mechanistic models, we believe, is more difficult and complicated than for physics-based models, for which there has been a lot of VVUQ formalization in mathematics. Two, it's in its infancy, so there's not much out there, and we're trying to get stuff out there, and other people are as well, but we really need a lot of work to be done. And of course, this presents unique challenges, such as, can we reduce model complexity? Are the models identifiable, that is not over-parameterized? Is there a need for general VVUQ, not just context of use? And can we develop physiological-based models with a context of use in mind from the beginning? So that concludes my talk, and I'll turn it back over to Wei or Don.
Thank you very much.
Great.
Very interesting talk. Yeah, so I think we have time for questions. So Don, can you just move cursor a little bit, show the audience where the chat bubble is? That's where you can enter questions.
Yep, at the top of your screen, unless you drug it somewhere else, you had a series of tools. One of them looks like this green bubble. If you click on that, you can both enter a question and see other questions that people have asked. So we've had some really great presentations today. If we have some questions for the speakers, this would be a great opportunity to get their feedback. And Wei, maybe I'll let you kick it off with one of your questions.
Yeah, so while we're waiting for audience to type in questions, actually I have one coming in right now. This question is for Marc Horner. And how do you deal with the variable calcification in the iliac artery? I guess that's a question for Marc.
Okay, yeah, thanks, Wei. So they were extracting some information from the CT scan data. Now, I know the focus, and at least what was shown in the picture, showed variable calcification in the region of the aneurysm. I believe that information may have been also present in the scan for the iliac artery. I'd have to double check and see if the resolution was appropriate in that area to capture the calcification. But I know at least in the abdominal aortic region, they were applying that.
And for the iliac as well.
Okay. All right. Thanks, Terry.
Yeah, I have another question for you, Thierry. How do you take into account the material properties and specifically related to specific patient, kind of patient-specific material properties? How do you handle that?
Well, it was very important for the cerebral aneurysm side. So the way they were doing that is to extract a different material property from cadaver after or before the rupture of the cerebral aneurysm. And they were making different kind of measurement in order to get this material property. From there, this is the kind of material properties that they are using in order to do this kind of simulation. Another part of the work that they are doing that we were not discussing here is to be able to predict the risk of a rupture. And over there, they need to use some free structure interaction where the material property is even more important than in the case of what I was showing to the positioning of the right web.
Okay, so there might be some difference between cadaver properties and the actual patient tissue properties, and is there effort ongoing in that trying to quantify the difference?
Well, so far not. One of the way to do that is to do some calibration where when you've got some kind of 4D, so the 3D with the time component, the deformation of the cerebral aneurysm, and considering this kind of deformation, you can match some kind of a simulation in order to make sure that you reproduce the same deformation. That would be one of the way in order to get the right material property for this specific patient, which is living. But this was not used as part of this simulation. And the one that I was showing today for surgery planning or during the operating room, during the operation itself, was just considering some simplified material properties related to the age of the patient and the gender of the patient.
I see. Thanks, Thierry. The next question is very interesting. I think it's probably for Rick. Have any clinical studies been able to leverage this type of modeling? So have you seen those modeling submissions through and demonstrate clinical benefits? Question for
me. Yeah, so that is a great question, and FDA deals with modeling at all different types of levels. So it's not an easy question to answer. I will give you a concrete example, and of course I can't talk about anything that's ongoing. We do, like I said, foresee a lot more physiological-based modeling. We anticipate it coming in. One example is the MRI-compatible pacemaker that was approved in the last, 2 or 3 years, where there were no clinical tests done. It was all bench test and computer modeling. But it should be noted that the computer modeling was not really physiological. That is, they were modeling the benchtop set where they had the wire of the pacemaker in a saline bath, and we're looking at the fields generated, the heating around that from the magnetic fields. And that was a device that was cleared without any clinical tests to the best of my knowledge. And I'm not speaking on giving any official FDA view. My personal opinion is that it is very complicated and multifaceted. I think in the drug world there might have been some approvals of high-level, top-down, non-physiological models that reproduce clinical trials, but don't hold me to that. I think that's true.
Yeah, thanks, Rick. And so we all know that FDA also received lots of submissions with modeling data, but they may not directly related to clinical. However, it still demonstrates the safety and efficacy of the device, and it's widely used in the development phase of those products in industry as well. So
yeah, that's a great point. I think it can be used as a tool throughout, and not just replacing clinical trials. There's a great question here about general verification validation. I'm not going to come down on a hard answer for that. Maybe Pras will chime in, but we're working on a paper along with some colleagues in the UK where we're really trying to compare and contrast and see what the similarities and differences are between general versus specific context of use. Pras, do you have anything to chime in about that?
Can you repeat the question? It hasn't appeared on my screen.
Oh, it says, for Pras and Rec, how might a more general V&V activity be constructed, one that does not have a specific context of use?
Yeah, it's a very interesting question, and it's one that we're trying to investigate. We're debating how FDA's approach is very context of use and risk oriented, and the cardiac modeling field has generally taken a very broad approach to assessing models, trying to study the model in general and trying to look at validation of multiple different model outputs. And what we're looking at, though we haven't really come to any firm conclusions yet, is how much these two things can be brought together. And we suspect that with physiological models such as this, extra general validation can be used to complement context of use validation and gain a better understanding on how the model works, which hopefully will increase credibility.
Great.
Yeah,
well, thank you, everybody.
Just one last note, and I think Marc Horner just posted a note in the chat window that there is a paper that you can download. Talks about heterogeneous material properties. I guess that's specifically related to a question a little bit earlier about the tissue properties for a specific patient properties. So there's a paper you can download. And Marc Horner put it on the chat window. I think with that, and we are closed down, and now it's up to you.
Great. Thank you very much, Wei. So thank you, everyone, for our virtual webinar and learning with us as we explore this type of a way to deliver both MDIC content and state-of-the-art activities that our members and broader audience have been working on. We had some wonderful presentations today from Terry and Mark, from Rick and Pras, and I want to give special thanks to Wei for agreeing to be MC today. That was a new twist that we've added. So thank you, everyone. Of course, this will be recorded and archived on our MDIC website. Thank you very much. Have a wonderful afternoon.
Thank you, everyone.
Thanks, Don.
Thank you.
Yep. Thanks,
Thank
Don.
you.
Thanks,
And Wei. And everyone.
Have a great day. Yep, bye.
All right, have a good day.